due to some features such as salt tolerance, psychotropic nature, and also the high specialization in metabolic pathway in the meat environment. For example, the arginine deaminase pathway and the use of nucleosides. I would like to thank my colleagues and Prima program that supported this study. And of course, thank you all for your attention.
Thanks for your presentation, Doctor. And then our presentation is open for questions. If anyone had any sort of questions, please drop your questions in the chat box. Or else unmute your audio and shoot the questions to Dr. Golia Tabanalla so that she'll be answering your questions. Besides, we will also share the contact mail ID of Dr. Golia Tabanalla so that you can reach her through the mail. I repeat, if anyone had any questions, please drop your questions in the box. Okay. Okay, it's like no one has any questions. Thank you so much, Doctor, for your valuable presentation.
Thank you.
Bye-bye. Have a nice day. And the next presentation will be given by Dr. Stephanie Grahl from Germany. The title of his presentation will be Consumer Orientation During a New Food Product Development, the example of spirulina-filled pasta.
SI is a sensory marketing research institute, and my daily work relates to sensory profiling, product perception, and consumer acceptance studies. And today I'm very happy to introduce you to parts of my PhD research, which was about the food product development with which relates to my daily work at ISI. Today's topic is called Consumer Orientation During New Food Product Development, example of spirulina-filled pasta. Before I tell you more about spirulina and product development, I briefly want to introduce you to ISI, what we are doing and what our daily work life is all about. We are one of Europe's leading agencies for sensory marketing research and innovation consulting. Our marketing expertise, market research, and sensory knowledge sum up to unique sensory marketing tools. We have a worldwide partner network. 70% of our projects are conducted on international grounds. We are a service provider for top global players in the food and personal care industry, but also for other research institutions. Quantitative consumer testing and descriptive analysis based on standardized conditions are the heart of our work. We do consumer tests and also descriptive analysis. Consumer tests comprise taste and odor tests, application tests, for instance, for body care, or packaging and design tests. Descriptive analysis conducted in our Sensei labs mainly deals with traditional product profiling, discrimination testing, as well as rapid and dynamic testing. That's it for now regarding ISI and what we are doing. Now I want to introduce you to the microalgae spirulina and tell you a little bit about what spirulina actually is, in case you don't know it yet. Spirulina is a cyanobacterium growing under extreme conditions. These extreme conditions lead to the fact that cultures are predominantly pure and have a low risk of becoming contaminated with toxin-producing microorganisms. So what do I mean with these extreme conditions? The cultivation takes place in an alkaline milieu with a pH of 9.5 to 10.5. The water is saline. It contains up to 30 grams per liter of salt. And spirulina grows preferably at very high temperatures between 35 and 37 degrees Celsius. as you can see in the picture, spirulina has its name because it looks like these little spirals where the cells are like coiled up in these small conglomerates of cells. Natural occurrences of spirulina are the dry areas of the tropics and subtropics with correspondingly high solar radiation. It can be assumed that spirulina has been eaten by Aztecs for centuries. Around the 16th century, one can assume. The use of spirulina from natural deposits in Africa, mainly from the Republic of Chad, has been known since the 1940s. Commercial mass cultivation began in Mexico at the end of the 1970s, and the biomass was mainly used as a food supplement, as a CO2 scavenger, but also for human and animal nutrition. Spirulina is now produced worldwide. We will focus on the Asia-Pacific region. The microalgae biomass that served as the basis for this project came from Myanmar and China. Cultivation is driven by modern biotechnology and various farming systems. These developments have led to the possibility of cultivation in regions that cannot otherwise be used for agriculture. Cultivation systems are typically classified as open or closed systems. Let me briefly introduce you to these cultivation systems. In Asia, spirulina is predominantly cultivated in open cultivation systems like open raceway ponds, as you can see on the left side. In Europe, closed systems like photobioreactors prevail. These large open raceway ponds have the advantage of lower construction and operating costs, sunlight as an energy source, but also a higher capacity, just to name a few. On the contrary, it needs more space, it is more difficult to control evaporation, and there's a higher risk of contamination due to being open to the elements. Advantages of the closed systems is the controlled environment for pH, temperature, and evaporation. Also that one can have a higher cell density and one is less dependent on the climate. Negative aspects of closed systems are that there's a higher risk of overheating, a risk of oxygen accumulation, but also they are very much more expensive to build and to operate. While foods of animal origin contain 40% and more protein on a dry matter basis, plants, with the exception of legumes, contain only up to 15% protein on a dry matter basis. Spirulina, on the contrary, contains 60% protein. The sulfur-containing amino acids, methionine and cysteine, are considered limiting in plant proteins due to their insufficient amounts for human nutrition, but the methionine and cysteine content in spirulina covers 73.8% of the recommended daily intake of these amino acids. Besides, spirulina contains all other essential amino acids. It has also a fat content of 5 to 10% dry mass, and about 20% of this fat content is gamma-linolenic acid, which is a polyunsaturated fatty acid. The blue pigment fucocyanin has been attributed to antioxidant properties. However, it is unstable to heat and light. As agricultural land is scarce and further intensification of crop production poses several environmental problems, the low land use of microalgae compared to meat could provide an opportunity to produce nutrients without converting natural habitats into arable land. So, as I said at the beginning, spirulina is not dependent on fertile soil, because if it is grown in these open raceway ponds or in photobioreactors, they can be built basically anywhere. And the specific growth requirements, the rather harsh conditions under which spirulina can grow, protected against contamination. The possibility of contributing to Europe's increasing independence from protein imports by producing spirulina in their own country should not be underestimated. So, as you have just heard, spirulina has a couple of advantages, but products at least in our German supermarkets, only include food supplements, snacks, and sweets, drinks, some specifically for coloring. So far, there are hardly any stable food products, and if they are not established widely. Research objectives related to this project were twofold. We wanted to investigate the suitability of spirulina for innovative foods that promote a reduced meat consumption, but we also wanted to integrate spirulina into Western European eating habits. There were a few challenges that we had to tackle. First of all, the spirulina powder had a very characteristic algae flavor. It had a dark green to black color, and we were aware of a lack of willingness to try due to new phobia. After all, research was targeted at the question as to what should be put on tomorrow's plate in order to integrate spirulina in European eating habits. The research was conducted in 3 consecutive steps, comprising the examination of the production process of spirulina, investigation of different product ideas, and finally a tasting test with consumers from Germany, France, and the Netherlands. In order to answer the question what should be put on tomorrow's plate, we needed to come up with an idea how to process spirulina to bring it into a staple food. And we decided that high moisture cooking extrusion would be very promising. But to understand the process better, we needed to know how the processing influences sensory properties and also how much spirulina can be used in a process where usually or most often only soy is used. When we had the X-rays, we did an online concept test where we wanted to investigate the product ideas and figure out how promising these product ideas were. And also, we wanted to understand how familiarity influences acceptance. Lastly, we did a tasting test of various products with consumers, and we wanted to know which flavor was liked most, and also how much spirulina soy extract consumers accept. On the following slides, I put these 3 little icons for you to easier follow at which stage of this project we are, and you will realize all these 3 icons on the slides as a sign that this belongs to either step 1, 2, or 3. The aim of high moisture cooking extrusion is to produce a textured product with a meat-like fibrous structure from a powdery premix, in our case soy and spirulina and water. High moisture extrusion cooking is a continuous process where proteins need to unfold, cross-link, and align themselves to form fibers. During extrusion, proteins transform and interact so that a 3-dimensional network is formed. And the process can be influenced by varying process parameters such as moisture, barrel, as well as cooling temperature, screw diameter, and speed of the twin screws, as well as die geometry impacting pressure, shear, and friction. So as you can see here, you have this engine and then these twin screws that rotate and knead and sort of push through the mix of protein, powder, and water. And afterwards, the mass is cooled under pressure, and then the unfolded proteins align, and you get a product that looks like this in real. This is the machine at the German Institute of Food Technology in Quakenbrück in Germany. And you see the product that we produce, that is an endless string of a protein bar, so to say. And this only appears when it runs out from the cooling. And here's the closer picture of these products where you see it cool down and it turns darker, and the lighter green product is straight from the extruder. As I just said, the process can be influenced by varying process parameters. And by means of a multifactorial design of experiment, it was systematically determined which extrusion parameters significantly influence the sensory perception of the products. And with this design of experiment, we simultaneously varied the different process parameters in an incomplete experimental design. We had these 4 process parameters in these 3 factor levels. Varying spirulina content, temperature, screw speed, and moisture. And we also looked at a batch effect of day production day 1 and 2. So in total, we produced 16 products to estimate 162 potential process parameter combinations. Once we had the 16 products, we screened 12 panelists and trained them according to ISO and DIN standards in order to develop suitable vocabulary to describe spirulina soy extrudates. The panel sessions took place at the University of Göttingen, and the aim was to find non-hedonic terms that are unambiguous, and if there was uncertainty what the terms meant, we worked with different references. And it was important that the panelists were able to differentiate between intensities. So at the end, we had a description of the products and the quantification of the intensities of the extrudates. Here you see the results of the sensory panel measurements. It is a PCA plot that contains the attributes that were necessary to describe the extrudates. And the attributes are accompanied by capital letters, and A stands for appearance, O for order, F for flavor, M for mouthfeel, TX for texture, and AT for aftertaste. And you also see highlighted or marked with stars the process parameters that we varied across the factor levels. The gray dots represent the 162 estimated products that would have been possible to produce, but we, as you know, had only 16. And when we circle the products that have the same spirulina content in common, you see that the products with a low spirulina content are located to the left, and the products with a higher spirulina content are located to the right. And we find that the more spirulina, the darker the color, the earthier the aroma, the stronger the algae flavor, and the more intense the umami flavor. We also find these products to be less elastic, less fibrous, and less firm. Besides spirulina content, moisture is most influential towards sensory profile of extrudates. Texturization is more distinct with lower moisture but higher screw speed and temperature. So the more moisture, the juicier the extrudates, the softer and also the more moist in appearance, but less crumbly. And on the opposite, the lower the moisture, the more fibrous, layered, and firm the extrudates turn out to be. So we can learn from that it is advisable to lower the moisture in order to get a more distinct texture. Now that we knew that it was possible to substitute some of the soy with spirulina, we had to answer the questions how we would proceed with the extrudates and how to process them into actual products that were possible to present to consumers. So we interviewed 4 food experts and present them with different ideas that we generated beforehand. And Christine Knufmann, Jacqueline Amefalla, Sebastian Lege, and Professor Thomas Wilkes agreed that it would be necessary to process spirulina in substantial amounts, that it would be advisable to keep the product simple in order to reduce concerns. They all agreed that the color would be rather difficult for consumers to accept, and also that the taste of spirulina shouldn't be hidden. As you just saw, I presented the experts with different ideas in the round circle on the right-hand side, and there were various ideas that we discussed, but we ended up with these 3 products that were most promising, and that was spirulina pasta, spirulina sushi, and spirulina jerky. These 3 products were also chosen because they follow different concepts. Pasta would reflect familiarity, The sushi was chosen because there's a flavor pairing of algae surrounding the sushi and algae inside. And due to the dark color, the spirulina jerky was decided to follow up upon because it also follows the idea of extruded products that are usually produced to make meat replacement. So we had these three products and combined them with three benefits, i.e. health, sustainability, or innovation. And we did an online study where these three pictures in combination with each one of these three benefits was shown to 1,035 consumers from Germany, the Netherlands, and France. And we asked for acceptance, but also wanted to understand how familiarity with each one of these categories would impact the liking. Not only did we ask for overall liking and familiarity, but also for the degree of novelty of the products, the interest, and also expected flavor liking. We let consumers rate these variables on a 9-point scale. And here you see the estimated mean values that we got for each one of the dependent variables for the different products and the three different benefits. We found that there was a significant product effect and that pasta was liked best. That's the black line. Also, benefits differed except for novelty, because as you can see, the lines cluster together in the upper part of the plot. And we found that the health benefit is most interesting to consumers. So generally we see that the degree of perceived novelty is comparably high, that marketing strategy of jerky should be innovative, and that the pasta is most promising and most liked by the consumers. We also found an interaction effect. That means healthy pasta was more interesting than sustainable jerky. We found differences in familiarity with the product category. Jerky was least familiar, sushi was in the middle, and pasta was most familiar. And the product effect that I already mentioned that we found, we wanted to understand better and figure out how we can explain this in more depth. So we put familiarity into the model, and then we found that the higher the familiarity, the higher the liking, and the actual product effect is weakened when familiarity is taken into account. That in turn means that spirulina-filled pasta was liked so much because it was so familiar, and jerky, since it is hardly known, was more difficult to imagine and received lower liking ratings. From the concept test, we knew that pasta should be followed up upon, and from the first study, the sensory panel analysis, we knew that spirulina tastes distinctly of algae and rather earthy. So we had to come up with concepts how to develop recipes in order to properly accompany the spirulina flavor, and we used flavor learning because we know that spirulina contains glutamic acid, and also cyanobacteria may contain geosmin. So we added tomato, which also contains glutamic acid, to the spirulina-based filling as a first recipe. A second recipe contained spirulina combined with beetroot, because we know that it is geosmin that gives beetroot the earthy flavor. So we sort of combined the two earthy aromas together. And as a second approach, we used masking, because we know that geosmin in an acidic environment turns into agosmin, and that is an odorless and flavorless compound. To investigate which flavor fits best with spirulina and how much spirulina is acceptable, the three flavors were combined with three different extruded contents in the pasta filling. So we had the tomato, beetroot that was added with ginger, and the lemon with basil in combination with each one of these three extruded contents. And then we produced the fillings and the pasta and had 350 consumers tasting the products. in an incomplete design, so 6 samples per person were served. And as you can see here, the pasta was served plain and without any sauce or seasoning to let consumers evaluate liking of pasta and its filling unaffected by other flavors or sensory cues. Here you see the results of the tasting test. We find that with mean values between 6 and 7 on a 9-point overall liking scale, consumers generally like the pasta variants. We find that lemon is liked most, tomatoes in the middle, and beet is liked least. And we also see that acceptance decreases with increasing spirulina content. We also find that higher spirulina content calls for a recipe optimization, particularly for tomato, because we assume that tomato fits least due to this steeper decrease in liking with increasing spirulina content. So what can we conclude from this? We found that familiarity of the category facilitates acceptance of novel ingredients, and we would recommend to integrate spirulina into well-known foods. We also find that early consumer orientation is a very good way to establish ideas and to test ideas, and at the same time let consumers evaluate benefits and recipes. We also find that it is important to consider a fit of sensory perception, concept, and context, and we would recommend to emphasize the benefits of spirulina. After all, running consumer tests with international focus is our daily business, and we provide highest quality in our excellent partner network. If research that you just saw is of interest and of relevance to you, then please don't hesitate to get in contact with us. We at ISI are absolute product testing experts. We can react very speedy, agile, and fully client and challenge oriented. Our processes are very automated, and so research can be done fast and more cost effective. And last but not least, we don't come alone. We have a very large network that we cooperate with, and you could benefit from our research and innovation ecosystem. I thank you very much for your attention and your interest. And in case of any comments,